Today we'll be learning about mutagenesis, and we'll have a live demonstration of how to induce point mutations into plasmids. So let's begin. Our bodies are very complex. There's so much we don't know about them. But we do know that what controls the cell's behaviors that allow our bodies to work is stored in our DNA. Each human has so much DNA, and each DNA has as much information as a high-quality picture. However, unlike changing one pixel in a picture, like the Mona Lisa, which you wouldn't even notice, sometimes a single letter change in DNA can cause life-threatening diseases. Because of this, our cells are really careful to not make mistakes when they're copying DNA. However, when our cells make errors and the result is a life-altering mistake, we might want to change it back with mutagenesis, the process of changing DNA. We will be focusing on how errors with DNA replication occur in plasmids. Plasmids are small, circular DNA fragments in bacteria, not part of the main DNA. They have genes that give bacteria superpowers that help them survive. In order to understand how to induce point mutations into plasmids, we'll need to review what DNA replication is first. So DNA has two strands that lock with each other perfectly, like a zipper. To copy it, DNA gets untwisted, like how a zipper gets unzipped, and the matching strands get added to each side. This is done with DNA polymerase. Our cells know where to start this process in our DNA. However, in labs, we have to use short strands of DNA called primers to match existing DNA and let the DNA polymerase know where to start replicating. This means we can change DNA in plasmids by using our own custom primers. As long as most of the strand matches, DNA polymerase will still go on with replication. Please note that even though we only have 4 letters drawn on our strand, the strand should usually be around 20 to 30 letters for the primer. Even though our primer doesn't perfectly match the correct matching sequence, it will still get incorporated into the new strand, changing the original function of the DNA. Replication then keeps happening, but the new strands are replicated more than the old strands because the primer matches with the new ones better. After replication, something called a restriction enzyme cuts up the old DNA, leaving behind only the new, modified DNA. Now let's meet our researcher and see how she does this in the lab. So why don't you start off by telling us about yourself?
Okay, so hi everyone, I'm Katherine Zhang. I am a fourth year student at the University of Virginia, and I'm double majoring in biology and biostat.
Let's begin the experiment. So this is the setup. Here, Katherine is adding all of the items listed above, which includes double distilled water to hold the reaction, the original DNA, the building blocks of DNA, and the molecular DNA replication tools. Now she is gently mixing the solution by pipetting it up and down several times. Lastly, she's going to add DNA polymerase, and here she is mixing it again by pipetting the solution up and down. And here's the final solution. Now she's going to run PCR by placing the tube into the PCR machine, and the instructions for the PCR cycles are in the top left. Essentially, the instructions are telling the machine to change the temperatures up and down for multiple cycles. So what's actually happening right now? Polymerase chain reaction, or PCR, is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, so that Katherine is able to amplify her DNA sample with her primer. Next, she's going to add DPNL restriction enzyme to the amplification reaction, and she's going to mix it and then spin the reaction mixture in the centrifuge for 1 minute. Lastly, Katherine is going to immediately incubate the tube at 37 degrees Celsius for 1 hour to digest parental. We just demonstrated one way to edit DNA, but there are many ways. CRISPR-Cas9, which was only recently discovered, is another tool for mutagenesis. Now we're going to have a Q&A to learn more about our researcher. How did you get into the research?
So I'm a bio major, right? So I start taking a lot of biology classes, and then we talk about all the cool techniques in class. So that really made me want to try it out myself. So I just reached out to Professor Leventhal, and then he got back to me. We set up a time. He told me more about his research, and I just really like it. So yeah, that's how I start.
How do you do your research and what gets you excited?
Yeah, I think I really enjoy doing the research because I enjoy like exploring the unknown field. I know research is not always very smooth, and sometimes you don't get a result that you want. But that doesn't mean the experiment is not working. And also, all the people around you will be like super supportive and super helpful. So for me, I just really enjoy like getting a project and brainstorm on a topic and carry out an experiment, whatever the result is. People around you will be very excited. They will try to help you, like try to give you some input that they have, and maybe you'll get a really good result, and then it's just like even more exciting, and then you feel a very strong sense of accomplishment.
Great, thanks. What's your specific reason for using mutagenesis?
Yeah, so for this project, I'm trying to compare 10 proteins. All of them are basically the same, but only with a slight different point mutations. So that means like it's all the same, but just for one amino acid is different. In this case, mutagenesis by PCR is very useful because it's low cost and it's easier to carry out.
And last question, how can people learn more about DNA and get involved with research?
I would definitely encourage people to take a biology class, especially a genetics class. And if you really enjoy it, then you could research like all the work of the professors in your school, and then maybe one of them, like his work will catch your eye, and then you can reach out and just introduce yourself. Like say you're interested in doing research and you're interested in the field, and then hopefully you can start your research from there.
Okay, thank you so much, Katherine.
Thank you.